DNA engineering consists of combining fragments of existing DNA sequences to generate new ones. So it's a bit of copy and paste of DNA. And in this example here, I'm showing the construction of a new circular DNA sequence from two existing ones, and researchers have access to tools that can, for example, cut DNA at particular places, so they can cut fragments of sequences and then, for example, join them together to make a new sequence. So there are many ways to do this. There are different techniques, but ultimately it all comes to like combining existing DNA fragments to generate new ones. The issue is that we don't have an open standard to document the steps we follow to go from existing sequences to new sequences. And proprietary software with which you can do this kind of thing doesn't really allow you to export the history in a useful way that you can use outside of this proprietary software. So OpenCloning, it provides both a data model to represent these sequence provenance, so the steps we've followed to create a new sequence from existing ones, as well as a web application with an intuitive user interface to work with it. So very briefly, the idea of the data model is that every sequence comes from a source, and this source can be an external import. So the user provides a file, or the user requests a sequence from a repository. For example, AdGene is a popular repository for sequences of plasmids, or for example, the user retrieves a genome sequence from the NCBI. So that is the provenance or the source of that sequence. But the source of a sequence can also be a manipulation that the user made. So they took some existing sequences, they did a manipulation in the lab, and they produced a new sequence. So you can see how connecting every sequence with its source, we can construct the history of a given sequence that we engineered in our lab. So that's basically what I want to say. The other thing I want to say is that apart from sequences, the users of eLabFTW who are doing molecular cloning, they will typically use something that is called a primer. So there is also support for this. And so a primer is a short fragment of DNA. So if you remember from your high school textbook, the way that DNA replication occurs is that you have a helix that is complementary. There is something in the cell that happens that separates the two helixes, and then the cell fills the gaps with complementary sequences to generate two identical copies of the original one. So PCR is something that kind of hijacks this mechanism and allows researchers to design short sequences that we call primers to amplify particular subsets of the DNA and then use that for their experiments. So this is something that is really important for people who are doing cloning, and there is support for this as well. So we can represent both the sequences that represent, for example, the sequences that researchers have in a cell line on a microorganism, or for example, as a standalone sequence in a test tube, but also we can represent these primers that they use to take fragments from those sequences. So with that, just going to briefly list the things you can do with eLabFTW and OpenCloning. So today I'm not going to show a lot of what OpenCloning can do, but only what the integration allows you to do. And then if you are interested, you can have a look at the documentation of OpenCloning where everything else is shown. So using this integration, you can load sequences and primers from eLabFTW resources. So for example, if you have a resource in your database that has a file that contains a sequence, so this will typically be a gene bank file, FASTA file, DNA files. These are common file formats that are used to store sequences that your users will be familiar with. So you can load this sequence into the website, you can use them to design new sequences, and then save those to the database of eLabFTW as a sequence file, as a JSON file that contains also the history, not only the final product. And also there will be links between a new sequence and the sequences in the eLabFTW resources that were used. And then you can reload it. and there will be a way to represent cell lines and strains, but this is something that is coming in the documentation that hopefully will be live soon. So now that I've said this, I can show you how it works. So if you have an instance of eLabFTW where you have the tool enabled, it should be here in Tools, DNA Cloning. So for example, here I have a bunch of resources in my database. And let's take, for example, this plasmid here. So this is a resource that, as part of its attached files, it has a sequence file here. It's called pdonor221.gb. This is a gene bank file. So if I come here to the OpenCloning window, I can click on import from eLabFTW. And then here I'll be prompted to select the category. So here, of course, all the categories that I have are related to DNA, but if you have, microscope, whatever, it will also be listed here. Then we type the name of this resource that we know, and then we can choose the sequence that contains the sequence. We can choose the file that contains the sequence to load it. So we pick it, and when we click on submit, this will load the file into the interface, so we can start cloning. We can start planning our experiment here. Another thing we can do is we can load sequences from files, so the user can just drop a file here and it will be loaded. And also we can retrieve sequences, for example, from a repository. So that's what we can do now. For example, we are going to get a sequence from a repository, in particular AdGene, which is a popular repository for plasmids. And this will make a request based on the identifier we provided and get us the sequence. And then the users can also explore it in a sequence editor here, but all this is covered in the videos that show how to use OpenCloning in general. So let's say now we have loaded a sequence that doesn't exist yet in our database in eLabFTW. So let's save it. So for that, we click on this floppy disk icon, and we can change the name, and we can select which type of resource we want to save it as. So in this case, we just want to save it as a plasmid, and we click on submit. And you will see that it gives us a notification that this has been created successfully, and it has switched from being blue to being green. And instead of the floppy disk, now we have this link that it's telling us that it's stored in eLabFTW. So now if I go back to my resources, I will see that this new resource has been created, which the name we gave it, and it has a gene bank file. So this is a generic sequence format for storing sequences and features within those sequences. And it also has a JSON file that has the history of this particular plasmid. So this history is not very long. We just load it from a repository, but if we look at it, we can see in the JSON that it tells us that the source of this plasmid is that it comes from AdGene, and it provides us a unique identifier of this plasmid in that database. So it is a way to trace back the provenance of these sequences. So then now, for example, let's say that the user wants to make a PCR, like we said before, starting from this plasmid. So they will have to design primers, so they will have to design sequences that allow them to extract a subpart of this plasmid. So you can do this in the interface. So you can say, okay, I want to use this to make a PCR, and I want to design primers for this PCR. And the purpose of these primers, in this case, we're just going to amplify. We just don't want to do anything else. So we select that, and we can select within the sequence what fragment we want to amplify. So in this case, this fragment of the plasmid. And then we can put some settings, etc. But we're going to take the basic ones. We click on Design Primers, and OpenCloning will design 2 primers for our example. So then we click on Save. They will be put in the PCR, and then we can do the PCR, we get the product here, and let's call it PCR product. And then we can save it again to eLabFTW, except this time, because we are using primers to create this, it will also allow us to save those primers. So for example, we're going to save this as a linear DNA, and it's asking us, do you also want to save the primers to the database? So we will say yes. We will save as primers, click on submit, and then if we go to our database, now we will see that we have created the PCR product, the two primers, and then when we go to the PCR product, we can see that the product is linked to its to the resources in the database that were used to produce it. So it has a link to the two primers and to the sequence that was used as a template for the PCR. And I think that's basically all I wanted to show. You can also create the primers directly yourself, so they don't have to be created through OpenCloning. You can create a primer directly in eLabFTW just by clicking on Create. Select the type you have associated, give it a name, call it my primer, and it just has to have a field that is called sequence. So I'm just going to put some random DNA sequence, and then now I should be able to load it from here as well. So I can go to primers, import from eLabFTW, a primer, my primer, and you can also import it into the session. So that's basically like a very, very quick overview of the features. And, I think there will be good documentation for this soon. Have a look. If you have some questions, please don't hesitate, to ask for me. I'm very happy to, talk to you if you're thinking of implementing it in your institution. If you have some users that are wanting to get started with it, I'm happy to give a presentation for them to kind of like help them migrate from what they use now to using this. So yeah, just I'm very keen to help you transition from whatever solution you have now to using OpenCloning. So thank you.